Good afternoon, everyone. Thank you for joining this session. So, I'm Haritha. I'm a postdoctoral researcher here from the University of Copenhagen. Today, I'll be sharing our work about how brain responds to extreme heat. Generally, heat stroke is often discussed from a cardiovascular and rehydration perspective, but brain actually is one of the first organs to fail under extreme heat. So as the planet warms, understanding how heat affects the brain is becoming increasingly important, both scientifically and from public health perspective. So to understand why this research is relevant, we should look at the heat-related mortality rate. Thoughts from yesterday and today have covered these topics about burden of heat and health effects under extreme heat. So I won't repeat the same numbers, but I would like to highlight that last year alone, more than 60,000 people died in Europe due to extreme heat. And this number will rise with each passing summer due to prolonged heat waves. As the planet warms, heat waves are becoming more frequent and intense. What is a heat wave? Heat wave is prolonged duration of abnormally hot temperature or hot weather. Sometimes it lasts for days, sometimes it lasts for weeks, and sometimes it lasts for months, where the minimum and maximum temperature stays higher for that particular region. So mortality data clearly shows heat now is one of the deadliest natural hazard. And despite these statistics, we don't fully understand how heat affects the brain. And this exactly is the focus of our research. Our body strictly regulates the internal temperature around 37 degrees Celsius through a process called thermoregulation. But heat stroke occurs when the internal temperature increases more than 40 degrees Celsius, which overwhelms the thermoregulatory system and leading to its failure. That causes a significant damage to brain due to reduced blood flow and oxygen. One of the earlier symptoms of heat stroke are neurological, like confusion, agitation, slurred speech, unsteadiness, loss of consciousness, and in some cases, even seizures. So these initial symptoms are just not discomfort, they are actual brain dysfunction. So this is why it is important to study what happens to brain under heat. But the repeated exposure to the heat without adaptation is harmful. So how can we safely study the brain under heat? So interestingly, the heat biology has this fascinating aspect where body can adapt to the repeated exposures through a process called heat training or heat acclimation. People like athletes, people living in hot climates, outdoor workers, they all go through this process called acclimation. So what are the physical adaptations to this heat training? So they become efficient in sweating, their cardiovascular stability becomes more enhanced, and also they have better thermoregulation. For example, a person from a cold country visits a hot country, like for the conference or something. So suddenly, initially they feel a lot of discomfort, but over a week or over a couple of weeks, they'll get more comfortable because their body adapted to the hot environment. They have some physiological adaptation happen. Initially, you could see they start sweating a lot. But what happens to the brain? Can we train the brain to get more resilient to the heat? So this led to our central question of the research. So our main question was what happens inside the brain when it gets too hot? And the second one is, can we train the brain to become more resilient to heat? So to answer these two questions, we need an experimental model where we can actually visualize the brain in real time when there is an increase in temperature. And the second one is we need an organism where it doesn't thermoregulate. That means the internal temperature reflects exactly that of its environment. So these two criteria best fit with the zebrafish. So let me introduce you to the zebrafish in detail. Zebrafish are small freshwater vertebrates, and they are used widely across all research fields, not only in neuroscience, but also cancer biology, immunology, and so on. So these are the adult zebrafish. They grow up to 1 to 1.5 inch long, and these are the larvae. You can see this. They have this transparent body and transparent brain. They are size of just a rice grain. And as I said, they don't thermoregulate, which is perfect for us to do heat experiments. And they're easily manipulated genetically. Here in this case, we have a green fluorescent protein attached to the brain. You can see these tiny green heads under the fluorescence microscope. This is the brain. So whenever there is an activity in the brain, we can see a flicker of this fluorescence happening. So if we zoom in, so this green part is the brain. So basically, in conclusion, zebrafish is the best model to study the heat stroke. And can we simulate the heat waves and see heat stroke-like symptoms in the zebrafish? So yes. What we did was, generally zebrafish are maintained at 28 degrees. But for our experiment, we started increasing the temperature to a certain point where they kind of collapse, and then we bring back them to normal temperature where they recover. If you look at it, in this first video, you can see at 28 degrees that it is swimming normally. But as the temperature increases, you can see the swimming got faster. It looks like kind of agitated, which kind of reflects the human symptom where they get irritated and stuff. And as the temperature increases even further, it starts swirl swimming, which is not a common or healthy kind of swimming. So here this behavior reflects the disorientation in human heat stroke symptoms. And as the temperature increases further, reaches a critical moment where the fish cannot cope anymore and it loses its response. It's similar to becoming unconscious in human heat stroke symptoms. As soon as it reaches this temperature, we bring it back to 28, and the normal swimming is resumed. You can see this larva is swimming normally. The thing is, so we focus on this particular, we focus on this particular 41 degrees, like this critical temperature where it loses the response. This is our like readout. So we are still establishing the heat stroke model here. So now we see behaviorally, it shows all the behaviors that reflects the heat stroke symptoms in humans. Now we want to see what happens in the brain. We use a similar protocol where we give the heat ramp and we look the brain under the fluorescent microscope. And here you can see the activity. So this flickering is when brain is active, you see the flickering, and this each line you can see here in the graph. Battery is not working. It is the neural activity. So initially, when the temperature is normal, there is a normal activity, and then slowly the activity becomes more frequent, and as the temperature increases further, the brain enters a phase of like silence. It's trying to protect itself, and if the temperature doesn't go down there, from there, it kind of enters another critical phase called brain shutdown. Here you can see this huge fluorescence, which is totally abnormal. Normal healthy brain doesn't show such phenomena. And when we bring it back to normal temperature, it resumes its normal activity. So here we use this peak where we see huge temperature, that temperature as a readout for the behavior experiments. So now we have established the heat stroke model in the fish where we can see both behavioral patterns as well as the brain activity. Now we want to see if we can train the fish or acclimate the fish to the higher temperatures and see if it imparts any resilience. What we did was, it's a very simple protocol. We grew fish embryos at 28 degrees and another batch at 33 degrees for like 5 days. And then we did the experiments on the 5th day, both behavioral and brain activity. So For behavioral experiments, we look at the temperature where they lose the response, and for the brain activity, we look at the temperature where we see this huge fluorescence. And the results are like striking. Here you can see the control fish loses its response at 40.8 degrees Celsius, whereas in the activated fish, you can see they lose a response at 42.7 degrees Celsius, which is like almost 2 degrees difference between the two groups. It's quite significant. From this, we can say just the mere 5 days of acclimation has imparted behavioral resilience in this fish. So now what happens in the brain? Does the resilience also extend to the brain? So we looked at the brain, again between the same control and acclimated fish, which are like trained for like 5 days in the heat. We do the same heat trap experiment, and we looked at the control as well as the acclimated fish, and we saw that control fish brain shuts down at 39.5 degrees, whereas the acclimated shuts down at 42 degrees. Again, there is a significant difference between both groups. This again shows the mere 5 days acclimation imparts nice resilience even at like brain level. So now we want to see this is like whole brain level. So now we want to see what happens at the cellular level, what molecular changes are happening at the cellular level. So let's see, this is a cell that any cell that has so many proteins in them, which helps them to maintain normal health and normal activity. In brain, we chose these two key players, which help the brain cells to maintain its optimal activity and health. So we'll take what happens to these proteins, their expression and their activity. So initially, we started with these sodium potassium pumps. We looked at their expression and we saw there is a significant decrease in the expression of this pump in the acclimated fish. Simultaneously, for the leak channels, I did pharmacology where I blocked the channels. Here, if you see, this is a control where you can see there is a difference where the brain shuts down at different temperatures. You can see the clear significant difference between them. But when I add the blocker, you can see both the brain shutdowns at the similar temperature. So whatever resilience was imparted by the acclimation was completely vanished when the blocker was added. So we hypothesized probably these changes in these molecules. Of course, there are so many other molecules. We didn't look at all of them. So far, we looked at only two, and we saw drastic changes, at least at this level. So in conclusion, we can say that we established a nice zebrafish heat stroke model where we can study heat stroke. And we showed that acclimation imparts resilience to both behavior as well as the brain. And we identified these molecular pathways that changed after heat acclimation, and that might contribute to heat resilience. So these pathways may one day guide treatments that help protect vulnerable population during extreme heat. So I'd like to acknowledge my lab, my PF Florence Karman, and my lab members, especially LSCO, who contributed significant amount of work here, and of course the funding, the Kavya Foundation. Thank you very much.